All right, let's jump right in. You might look at the vestibular labyrinth and see two kinds of hair cells and think, okay, a little bit of biological redundancy makes sense. But that's not it at all. This diversity is actually a brilliant design feature, an evolutionary masterpiece, really. And it's all engineered for one thing: incredible speed and precision.
So, for this deep dive into the nuts and bolts of our balance system, our go-to source is the Anatomy of the Vestibular System, a comprehensive clinical review. It's absolutely packed with the kind of detail we're going to get into today.
Okay, here's how we're going to break this down. First, we'll talk about the problem of speed, why this system has to be so ridiculously fast. Then we'll look at the two cells at the heart of it all and their different strategies. After that, we dive into the star of the show, the calyx synapse solution, and we'll wrap up by connecting all this amazing cell biology back to the clinic, looking at what it means for our patients.
All right, first up, the core challenge. The vestibular system doesn't just sense motion. It's the engine driving some of the fastest reflexes in our entire body. And it's all designed to do one thing: keep our view of the world rock solid, completely stable.
And that brings us to the big question, the one that really drives this whole story. How does the vestibulo-ocular reflex, or VOR, pull off its magic? The reflex that keeps your eyes locked on a target while you move your head. It has to react in less than 10 milliseconds. I mean, how is that even possible? What kind of biological machinery can move that fast?
Well, here's the thing. Your standard issue neural transmission, with chemicals floating across the synapse, it's got a built-in speed bump, a delay. It's just too slow for this job. So to hit that sub-10 millisecond target, the system needed a workaround, a kind of biological hack to completely shatter that speed limit.
And that special solution? It starts right at the source, with the sensors themselves, the hair cells. See, nature didn't just phone it in and make one type of cell. It engineered two distinct types, creating this incredible division of labor from the very, very beginning of the process.
Okay, so this image just shows the basic mechanism perfectly. It's the same for both cell types. As that hair bundle gets deflected, these little tiny filaments, they're called tip links, they literally get tugged on. And that tension physically pulls open ion channels. It's a direct mechanical system. No complicated chemistry, no waiting around, just pull. And that pull lets potassium and calcium flood into the cell, which of course depolarizes it. It's just mechanotransduction in its most pure, elegant form. But this is where things get really interesting and the specialization kicks in. On one hand, you've got the type 1 hair cell. It's shaped like a flask, and it's almost completely swallowed by this huge nerve terminal called the calyx. Then you've got the type 2 cell, which looks a bit more, normal. It's cylindrical, and it hooks up with a bunch of smaller nerve endings called boutons. Just looking at their shapes, you can tell they're built for completely different jobs. And this table just lays out that division of labor so clearly. The type 1 cells, which you find more centrally, they're the sprinters, the high-frequency specialists. They have this unique potassium channel that lets them fire in these irregular bursts, making them perfect for encoding those super-fast transient movements you pick up with a V-hit. The type 2 cells out on the periphery, they're the marathon runners. They fire at a steady, regular pace, encoding those sustained, low-frequency movements, exactly what you're testing with calorics. And look at this diagram. It really gives you a sense of that specialized setup for the type I cell. See how that flask-shaped cell is just hugged, cradled by that enormous calyx terminal. It's an incredible structure. And this unique arrangement is the absolute key to a totally different way of thinking about synapses. It allows for not one, but two different modes of transmission, the classic chemical way and a much, much faster method we're about to get into. Okay, so let's zoom in on that special potassium channel I mentioned a second ago. It's called IKL. And this channel, it's really the secret sauce for the type I cell's speed. It's kind of always a little bit open at rest, which makes the cell membrane leaky. And because it's leaky, it has a really low electrical resistance and a super short time constant. So what does that mean? It means the cell can charge up and discharge lightning fast, tracking every little nuance of a high-frequency head movement without blurring the signal. Okay, so that leaky channel is part one of the solution. But now we get to the real heart of it all, the synapse itself. The calyx synapse. And honestly, it is an absolute masterpiece of biological engineering.
And this whole setup presents what you could call the calyx paradox. I mean, think about it. Why would you pour so much energy into building one gigantic nerve ending just to wrap around a single sensory cell? It seems totally inefficient, right? Well, the answer is just brilliant. It's because this structure isn't built for one kind of communication, it's built for two, happening at the same time.
Okay, so mode number 1 is the classic chemical synapse you see here. The type 1 cell depolarizes, it releases glutamate from these special ribbon synapses. That's your standard quantal transmission. It's reliable, it's steady, it's great for those sustained, lower frequency signals. It's the dependable workhorse.
But this is the game changer. Remember that leaky potassium channel we talked about? Well, when the type 1 cell gets activated, potassium ions rush out of it. And because the calyx wraps around it so tightly, it creates this tiny confined space. The potassium gets trapped, it builds up in that little gap, and this sudden accumulation of positive charge directly depolarizes the nerve terminal. No neurotransmitter needed, no vesicles, no synaptic delay. It's not chemistry, it's just pure physics.
And how fast does this happen? We're talking less than 100 times microseconds. That's basically instantaneous. It's about as close as you can get to a hardwired electrical connection, and it just completely vaporizes the delay you get with a chemical synapse. This is how the VOR is so unbelievably fast.
So what you end up with is just the best of both worlds, right? The calyx synapse gives the system the steady, reliable signal of a chemical synapse for tonic stuff and the absolutely breathtaking, almost electrical speed of this non-quantal transmission for those super fast signals that drive our reflexes. It's an incredible 2-for-1 deal.
Okay, so that's the beautiful cell biology. But let's bring this back from the bench to the bedside. What does all this high-performance engineering actually mean for the patients we see every day?
So let's just recap the key points. The type 1 cells are built for speed, thanks to that leaky IKL channel. The calyx synapse then uses a clever 2-in-1 system to achieve near-instant transmission using potassium. But, and this is a really big but, this amazing specialization has a downside. It comes at a cost. These high-octane type 1 cells, especially the ones in the central streola, are really high maintenance. They're metabolically expensive, they're super sensitive, and that makes them a prime target for damage from things like aminoglycoside ototoxicity.
And that really leaves us with a powerful takeaway for the clinic. The very same specialization that makes our vestibular system so incredibly fast also creates its biggest weakness, its specific point of failure. It's the reason we so often see those high-frequency V-hit results drop off first when there's autotoxic damage. High performance and high vulnerability, they really are two sides of the same coin.
So we've covered the hair cells and the amazing dual transmission of the calyx. Join me in our next explainer, the afferent system and vascular anatomy, where we're going to explore why the inner ear's end artery supply means that any single branch occlusion produces such a precise, anatomically predictable clinical syndrome. Thank you for listening to this series. To explore more topics about dizziness, vertigo, and imbalance, and to get resources for both clinicians and patients, please visit AustralianDizzinessClinics.com. And of course, please subscribe and leave a rating.